So, hello everyone again, and I want to wish you another nice weekend. Welcome here again to another Saturday live stream. I'm Oliver, Microbehunter, I call myself. And today, is February 14th. And for this reason, I decided why not put a beautiful flower under the microscope. Not only that, after 45 minutes, I do want to show you a completely different little surprise that I found in a water sample today. Of course, also under the microscope. I'll show you later. Okay, so a little bit, a background here. I got this flower yesterday from a colleague. Yeah, she gave many teachers Valentine's flowers. They were sold by the students. You probably already know that I'm a teacher in school. So students basically were running around the school building, distributing and handing out flowers that were bought. I think it's a very nice, very nice idea. And when I received one of those flowers as well, with a little thank you note, then I said, "Wow, isn't this nice? Yeah, I have something to put under the microscope again." And look at this here. There are pollen even in here. So I would also like to make a pollen slide today, and I would like to use some glycerin gelatin as well to make a pollen slide. And I would like to also try to make a cross section of the stem here. So we're gonna see again how successful I am. And then, as I promised you later on, I do want to show you a little surprise that I found under the water sample today. So, but before we really get started, it's a tradition that I say hello all around the world. Ah, yes, saluton al vi ankaŭ. Yes, it's a couple of words in Esperanto, one of my important hobbies that I have. It's from the Netherlands, Canada, okay, London, Rhode Island, Jerusalem, England, France, Poland, Austria, okay, LA, England, Greece, UK. That's one of the nice things is that somehow a little bit the whole world is a little bit happily united here today. Yeah. So I'm going to give you first, a little. I want to show you something here that I prepared already. If you look over here on the bottom side of the leaf, you notice there's something strange here, right? Yeah, and this actually comes off, and this is because I put some glue, some wood glue on here a few hours ago, and in the meantime it already dried, and look how nicely it comes off. So I've made there are a couple of other glue spots here as well. What I would like to do is I would like to put this glue spot under the microscope, and I did not look at it yet. But what we have over here, these are so-called leaf impressions, and I highly recommend that you also try this. Just make sure that the leaf is like here on this flower is reasonably smooth. If you actually try to peel it off, maybe you're lucky, then it will come off quick, easily. If not, then you just want to try another one over here. Yeah, and I'll be dissecting the complete flower. This one went off easily. I'll be dissecting the complete flower today anyway. And let's turn it around and let's have a look at the leaf impressions here, because they capture the surface texture of the leaf. So what glue did I use? As I'm going to show you, there are different possibilities, but there's one glue that I can recommend, and pretty much everyone's got it. It is simply regular white wood glue. That's it. I've tried also other types of glues, like for example Elmer's PVA glue, which you probably know that I've already used a lot before for making permanently mounted slides. But I found that sometimes those glues, they have a high surface tension. They actually start to make how do you call this, droplets, and they don't stay spread apart. So what I've done is the following. I'm just going to demonstrate this to you. Yeah. I just want to demonstrate this to you. I always want to use the bottom side because I hope to see the stomates. And then all you do is you kind of press here a little bit, and then you go like this, and that's it. And then you wait, right? And then it will become transparent. So this is regular, regular wood glue. Okay. Yeah. So it's really nothing special. It's water-based, and the advantage of this being water-based is, of course, that there are no organic solvents in there which might actually maybe damage the cells or whatever, right? As a matter of fact, look, there is another one over here. Look, this goes off easily. Look at this, yeah? And sometimes, if of course the leaf is very hairy on the bottom side, right, then of course the glue is not going to come off quite easily. But here in this flower, it's quite easily possible. Let's have a look at this here, okay? Again, I've done this already, of course, many times before, but not specifically on this flower. And no cover glass, no mounting medium, no nothing, okay? Just a regular dry mount. And let's have a look, okay? Oh yeah, I have to change again. And then we're going to start cutting apart the plant a little bit, yeah? Yeah. Those roses can have a lot of pesticide residues on them, I just read. Yeah, of course, but I won't eat them, obviously. And I do hope that essentially the pesticides, which are certainly on them, will stay inside the plant. But I will now do the following scope view, and here we go. I need to turn this up a little bit. Yeah, I've got DIC. I'm gonna just do the following. Let's just look at this under bright field, like most of you have bright field as well. And yeah, you can see this is the 10 times objective, and here we go. Yeah, it's some parts are a little bit blurry because it's not completely flat. Yeah, but maybe you can find a different place here which is a little bit flatter. Yeah, and I go up with the magnification. Yeah, I mean, I always consider it quite fascinating. Yeah, to see how many details you're able to see here, and what you're able to see the individual cells already, right? Those long cells, and again, that's just the impression, yeah, that is left on the glue. And so it actually shows that the surface of the leaf is not entirely smooth. And what I think is that those things, where are they? Those things here, they look very much like stomates, which are opening in the leaf, which allows gases like, for example, carbon dioxide to go in from the air and oxygen to go out from photosynthesis. So I really like this glue method because, yeah, it's very easy and it looks kind of nice as well, right? Let's go down a little bit again with the magnification and let's have a look at a few, at the other one over here. Yeah. So look at all those, yeah. Here, that's nice over here. These look very much like stomates, which are the openings in a leaf. Yeah, which allows the gases for gas exchange. Those here, and they're able to open and close. Can you show it in simple polarization? Okay, folks, I'll be honest with you. I mean, I don't have simple polarization, but you see the glue itself is not really polarizing. So what I can try to do is, in any case, I can try it with, I mean, I'll try, you know what, I'll try it because I'll try it in any case. So the question is how does this look under polarized light? That's the request. I've got here two regular polarizing filters, right? Linear polarizing filters. So I'm going to put one of them on the lamp, the other one on the glue, and then we're going to have a look. And I don't think that this is going to make look very nice. Okay, so I have to remove the polarizing prism from the top, and I'm going to put this here now here as well, and then I have to refocus, of course. You know what? I'm going to start with a low magnification here. I'm going to swing out, remove the condenser here. And let's have a look how this looks on the polarized light. I have to really turn up the light intensity here. I have to focus. Yeah. And now I'm turning. Let's see if I can go a little bit higher with the magnification. I need way more light. Actually, honestly, I'm surprised this actually looks good. I thought actually because the glue itself is not really polarizing or anything, it's amorphous. This is simple cross-polar. Honestly, pretty much anyone can do this. It's simple cross-polarizing. You just take a polarizing filter and it still looks nice. Yeah, it's just regular polarization here. I think I cannot go higher because then the objective is going to touch the filter here. Yeah. Is it possible to see live stomata open and close, by the way? I do not think that this is so easily possible because you'd have to take the whole leaf and put that under the microscope, right? Or you make such a thin section, cross-section, and then essentially without damaging the cells. At least I would not know how to do that. I mean, that would be really nice to see, to actually see the opening and closing of them. Unfortunately, yeah. Yeah. Would phase contrast make much of a difference? I have a problem with phase contrast now because there is no cover glass on here and the distance is very large. So what I'm going to try it anyway, because that's the whole point of the live stream. I like to experiment around together with you, also not always knowing exactly what to expect. Okay, so this was now cross polarization. Now let's try phase contrast, and the problem with phase contrast now is that I only have it with in high magnification. Where's this? I have to open it. Okay, and you know what? I need to focus it first before I can even do anything. Without focusing, it's not gonna work, right? So that's again bright field. I'm going to now put in, and I hope I'm not touching the glue. It works. So that's now with a 40 times objective in phase contrast. No, it's not phase contrast yet. I've got the wrong filter. This is now, let's open the condenser. This almost looks like oblique illumination a little bit. Yeah, that's phase contrast. Nah, doesn't look nice. It's almost a little bit oblique, and I suppose because the glue is not completely flat. Yeah. Yeah, it's also very high magnification. In any case, I wanted to show this to you because I think it's a very nice and easy, it's also probably not properly centered. It's a nice and easy way. Let's now do the following. Let's now go full color because this is now DIC, differential interference contrast, and here that's how it looks like. It looks very nice. Yeah. So this is yeah, just something that yeah, I like doing occasionally, especially when I have a new plant. You always try out different types of glues and different parts of the leaf and so on, and then yeah, just observing a little bit of what we're able to see. And yeah, so that is basically the glue impression. I'm going to move on because I do want to spend a little bit of time also with the rest of the plant here. And what I really want to do today is, yet again, like every year, I would like to make a pollen permanent mount. And yeah, I think several months ago, last year, I've also made one or a few of those. And they're also quite nice looking. And I was really happy when I got the flower yesterday. I looked at the... Oh, you don't see that? Where is this? When I looked at it, I was really happy. Wow, there's pollen on here. Yeah. It's a little bit out of focus. I have to focus it again. Yeah, so these are the answers, and yeah, this pollen. And I said, wow, those pollen, actually, I would like to have a look at. And in order to do this, I'm going to now use a slightly more, how shall I say this, advanced mounting technique. No, I do need my glasses. My eyes are becoming worse and worse. And why do I say advanced? Because pollen grains are quite sensitive. So if you use a mounting medium that contains a solvent, for example, it's not going to work. The reason is because the pigment, the yellow pigment, is starting to leach out and it's going to dissolve. So you want to use a water-based mounting medium. And there is one that is basically a standard for pollen, and that's glycerin gelatin. And if you want to know a little bit more about this mounting medium, then you've got to look a little bit last year or so where I also used this. I just want to show it to you how it looks like. Yeah, this is the... people wanted to know where I buy all of these things. Many of the things I bought from Brunel Microscopy. It's a company in the UK. And yeah, they've got a lot of stuff there. And that's glycerin gelatin for pollen. And you see it's kind of pinkish, and because they added a stain to it to make the pollen, to stain the pollen. Now, glycerin gelatin is interesting because it is a mounting medium that does not dry. It's not supposed to dry. It's water-based, but it stays semi-solid. Like a gel, it's a gel. So what I have to do now is the following. I have to take out a small amount of this glycerin gelatin. I have to melt it, and then I have to put the cover glass on top, right? So I have to actually heat it up. But if I heat it up too much, then the problem is then the glycerin gelatin breaks because the gelatin does not like a lot of heat. Now, instead of just heating the whole bottle, the whole jar, I do the following. I take out a small sample and put it on a microscope slide, and then I will melt it on my coffee warmer. Okay? Okay. Oh, that's a nice one. I got my microscope there, from Brunel Microscope, so with bright field, dark field, and phase contrast. Yes. Yeah. Vein impression of a dried dead leaf. Not a vein impression yet, but I did prepare already some leaf veins. Okay. So, but let's get started with this and let's have a look. Okay. So this is how it looks like. There is. If you smell it, it does smell like it has this typical hospital smell. You know, that's phenol. Phenol is a disinfectant, right? So it's So because otherwise bacteria and fungi would start to break it down and decompose it. I don't want that, right? But I'm going to use the spatula now to go in and to scoop out something. But every time when you go into a container, you actually want to make sure that it's reasonably clean, because I do not, even though there is a disinfectant in there, I don't want to contaminate it. So what I usually do is I take a little bit of this is just this alcohol, just ethyl alcohol, and I just disinfect it a little bit as well, simply because I don't want to go in with dirty tools into this little jar here. Okay, not that this is a huge problem or so, but it's basically just out of principle I do that. So what I'm gonna do first now, how am I gonna do this? I need to warm I'm going to warm this up first. Okay, so I'm going to scoop out a small sample from here. Okay, I've already taken one already, so I'm going to take a small sample here. Here we go. I hope that this is going to be enough. Yeah, I guess it is. I don't know. I'm just going to. Here it is. Okay, here, this one over here. I'm just going to give it a try, and there is a general rule that this should not be much warmer than about 40 degrees. Where is this? Here we go. I'm just going to put this forward here. I'm going to put the flour away for now, and I'm just going to heat it up here on my coffee warmer, right? And until it melts, right? So I'm just going to put it on here. You can see it here, right? And in the meantime, I'm also going to prepare a little bit the pollen. And the pollen I have to pick off using, or I want to pick it off using, a toothpick. Let's see how quickly this actually. Lots of pollen on here. Yeah, look at this. See this? On the toothpick, lots of pollen. Very nice. It's slowly melting. Cool. It's slowly melting. Am I happy? I need a cover glass. As well. Oh gosh, I only have 2 hands. The cover glasses are dirty. Uh-oh. I need to quickly clean it as well. Yeah, you see? It's nicely molten now. Okay, I've wiped the cover glass. I like to use round ones now. And let's do the following now. I'm losing my pollen. I dropped some pollen. Let's try it again. More. Lots. That's a lot. And let's do it like this. You can also do a different method. You can actually also dissolve the pollen first in a little bit of water. Put it on the slide and allow the water to dry. Okay, but I'm just going to do it the straightforward way here. The simple way. And now I'm going to simply drop a cover glass on top here. Oh, it says it's slightly out of focus the whole time. Yeah. And let's go here, up. And we allow it to spread. Yeah, actually I got the quantity quite nice. And yeah, I think, look, it really spread nicely now. And I'm gonna put it directly under the microscope. So I'm gonna turn this off and put it away. So let's have a look at some pollen now under the microscope. Yeah, it's not very warm either. It's nice. And here we go. Where is the scope view? Okay. And here we go. Just occasionally reading this. If you have questions, please say I'm at Microbehunter Microscopy, so it's easier for me to actually find it. Okay, it's a little bit out of focus. Let's see if we're able to get it into focus. Here we go. Okay. It's interesting. There's some stuff that's kind of shriveled up. I don't know what that is. Yeah. But here, these are pollen. You see, it's still liquid. It's still liquid. Interesting. So maybe it's not fully, hasn't fully cooled down yet. Yeah. Yeah, because you're just wondering. This I used regular wood glue. Okay, just any white glue should do. Okay. Yeah. So these are some pollen grains, right? And let's see. Maybe as the slide cools down, it should actually stop moving. Now, this whole thing looks a little bit blue because of the DIC. I'm going to use now again bright field. And what we should actually see slowly over time is this, that the pollen grains turn pink. And the reason is because they absorb the pigment, the stain that was added to the glycerin gelatin, to the mounting medium. So after a couple of hours, they should be actually nice and pink. Yeah, and slowly you can already see that they do slowly start to turn pink as well. You see, now it stopped moving, so now the mounting medium has cooled out completely, and this then means that it's turned solid. And usually after, I don't know, an hour or so, you can notably see that the pollen grains are quite pink, but you can actually start to see this already, that they start to absorb the stain. Yeah, so this is a regular bright field. Let's go up with the magnification here. And of course, you know what I want to do in springtime. I want to grow some pollen tubes, right? So to actually observe the pollen grains germinating, growing a pollen tube. That's possible, but requires patience to get the conditions right. Yeah. I wonder if you could get hold of some animal brain cells. I wonder what we could see. I mean, I do have some animal brain cells over here in my cupboard. Some was it a mouse brain cells or something like that? Not super exciting, but because you're asking for it, I can actually try to find it later. How's that? Please remind me. I'm going to forget. Yeah. How long would an algae sample last in a permanent slide using regular clear PVA glue? Well, I've got a couple of slides, if I only find them, that actually I've got now a couple of years. So nothing lasts forever. But I mean, what I've seen on very old slides is that over time the algae, the green pigment starts to bleach out and so on. But honestly, if it's dry and everything, it should last for a very long time. Yeah. So you see now they slowly start to turn pink. Okay. Another thing I want to try is the following. Let's do this. Let's do this. Let's do this. Okay. That is now dark field. Okay. Yeah, so also looks kind of nice. Yeah. Contrast could be better. Yeah. And again, DIC. For those of you who are not so much into microscopy technology, I've got a pretty nice microscope that's able to do DIC, and DIC basically gives you a little bit the impression as if the specimens, like the pollen grains, that they kind of look a little bit higher, taller, because it looks like a little bit like this edge down here is darker than the other side. Yeah. So, and that's allows you to increase the contrast. But this does not mean that it actually has that shape. That's really important. It's just an optical illusion, right? Yeah. And you can do some fancy other things like changing the colors around and so on. Yeah. 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 So. Pretty nice microscope. Yeah, yeah. For me, you see, I always say I don't need an expensive car, I don't need a fancy apartment or anything, or expensive holidays. But what I do want to have is I do want to have a nice microscope. That's just the many things in life that I can, how do you say, I can do without. But yeah, yeah. So just yeah. So that's basically the pollen, and we're gonna move on because I think the next couple of steps are gonna be a little bit advanced. And I don't know how successful it's going to be. Okay, so the first thing which needs a little bit of time is I'm going to do something really horrific, but I think it's better that I do that because after all we're making a YouTube video here as well, and I'm simply going to now do the following. Don't be scared now. I'm going to cut off one of these petals. Yeah, so and I'm going to cut it off here, and you cannot see it. I tried this a couple of times, and yeah, it's of course now cut open here. I'm putting it in here, and now look what I'm doing. I'm taking a stain, and because this does take a little bit of time, and I really hope that this works, I'm taking a stain and I'm putting the stain, in this case, I don't know, you choose the stain, maybe regular pen ink, fountain pen ink also works. Crystal violet is a very strong stain, and I'm putting it down here only. Okay? Only down here, and we're going to wait a couple of minutes. And what I hope to be able to see is that the stain is actually being pulled up by the vascular tissue into the petals, that we actually see the place where the water is able to travel. Okay, so you probably already know, I have not done this yet, but you put basically, people have been doing this for a long time. You take a nice white flour, white is important, otherwise you don't see the color. You put it in some stain, and after a couple of hours you're going to see the flour change the color, right? Because as the ink or the pigment is pulled up, it starts to stain the white flour. Nothing unusual. This is something that's, yeah, you like to do sometimes with kids and so on. In the kindergarten, you change the colors around and so on. But that is something that I have not done now because I just want to keep it as natural as possible. And because we're going to do the following now, I'm going to just take this. I'm going to put it under my stereo. Oh, I don't know if you're able to hear me. The microphone is moving. I'm going to put it under my stereo microscope. And we're just going to see if we're able to see already the movement of the stain. And I can actually already see it. Okay. Okay. Here we go. You can probably already see that deep in here. Yeah. Do you see this? Yeah, it's starting to turn blue. Also over here. Right, so that's basically the pigment is being drawn up. Yeah, so that is that's the thing that I actually wanted to demonstrate to you. Is it capillary action? And not only. Okay, this is called transpiration, and it is a combination of capillary action, very important of course, but also that at the stomates in the leaf, the water evaporates. So basically it creates also a pressure difference. So it's a combination of both a capillary action and also water evaporating at the leaves, and then this kind of the evaporating water causes again a hole, so to say, which is filled up by the remaining water. Yeah, so it's called transpiration. Yeah, this whole process, and capillary action is, of course, one of the important driving forces. Yeah? So, but that is kind of just something I wanted to show you. Over here, here you see it also, right? We're just gonna wait a little bit, but that is, and as a matter of fact, you can maybe even see over here. Look, if you actually, let's zoom in a little bit more. I've also just realized this myself. Look, you can actually even see the individual cells here, right? Huh? It's kind of interesting as well. Those tiny little things here, yeah, are the individual cells. So, okay, we're gonna just leave it. This one over here. I need to just check the other one if the other one is still fine. Be the little surprise thing that I've reserved for later on. I need to add a little bit more water here, if you allow me. Because I already prepared the slide. And just a second. Okay. I'm finished. Okay. It's a little bit a very nice welcome surprise that I found in a water sample, and I just wanted to share that with you as well. So let's have a look, further look. So that is basically one of the things here, and I will now go back to my desk view. Here we are again. Okay. Okay. Yeah, I'm just reading every now and when I'm making a little pause. Then I'm also reading the comments here. Yeah. So now the next thing is going to be a challenge. Okay. So I'm going to now. I want to cut now the stem here, and in order to do this, I need a very sharp. And I'm going to use a microtome again, and I'm just need a sharp. How do you say, little thing here? So I'm going to cut it over here like this. So yeah, turn it, turn. Here we go. Here we go, okay. I'm just going to put this flower back into the water bottle. So just that you see what I'm doing here. Okay, that's it, okay. Yeah. Yeah, by the way, in case you realized, I've got I got a new camera right now as well, and the background is also now I've replaced the wall in the background as well, so I think it looks a little bit nicer now. So that's how it looks like, and now the big challenge: how can I make a very thin cross section here? And I need a microtome, right? So let's clean everything away here. I just want to show you also, yeah. I'm going to use my microtome, which some of you probably already also know already. It's this one over here. Okay. And down here I can actually turn it. I have to turn it to correct direction. No, the other way. So. And by turning it, it raises and lowers this piston in here. I'm going to go all the way. And you see it kind of locks in place, so it gives you a very precise way of adjusting the thickness. Still a big challenge getting, yeah. So, okay, now I'm all the way at the end. And now the problem is now, how in the world am I supposed to put it in here, right? Yeah. And the instructions basically say that I have to use a carrot and make a hole in a carrot, yeah, and use the carrot as a specimen holder. And I said, I don't want to mess around with carrots. So what I've done is I've 3D printed myself those little things here. And the only problem is with different sizes. The only problem that I have now is that the diameter here, I don't know if I'm able to fit it in here. It does work. Okay, yeah. So this is basically the, way that it should go. And this is kind of very tight now in here. And this now allows me to put it in here, all the way in. And you see that it's kind of touching now the edge here on the side. It's not in the middle. And this means when I use a knife, the razor blade to cut it, that it gets support of the edge of this microtome, right? Yeah, so it's kind of stable. Yeah, we're gonna see. So I 3D printed those things here. Right? Yeah, so trial and error. Those holes in here, those two holes in here are there so that it's easier for me to take it out again, right? So that's the only thing. And this one over here, I also 3D printed, and it's a regular razor blade. Okay? So let's have a look. And yeah, usually, yeah, the problem that I just found out is that the stem is generally a little too thick. Yeah? Yeah, for this. So I'm not able to cut all the way through, and this is indeed a little bit of a problem now. So, yeah, so this kind of, it's a very thick stem. So what I'm going to do is I'm going to try to advance it a few times. 5, 6, 7, 8, 9, 10. And I will try to give it another cut. And you see that, yeah, it doesn't always work. There is a little bit of a tolerance, and now see, up, I barely was able, yeah, to get it. Yes, this one is already, and I have an idea, and maybe some of you, I'm not so experienced in that, but I have an idea. It would be kind of interesting to make a microtome knife with some kind of a piezo quartz which vibrates with ultrasound. I know that there are those really expensive kitchen knives that vibrate with ultrasound to make cutting easier. That would be actually a nice thing here also for a for a microtome knife. Huh? So, but yeah, I wouldn't know how to do that easily. Yeah. So this is not a very nice cross section. Not a very nice cross section, but we're just gonna take it in here anyway. As so often with these things, it's all trial and error. So, yeah, maybe this works a little bit better. Some people say you should not be going back and forth, but you should just try to slice it in one piece. Yeah. I say we try everything. Okay. You try to spread it as good as you can. So, and again, here we go. Sometimes you can also put it into a little bit of water. It also sometimes helps. And then you try to get it out. I'm just going to do it the easy and straightforward way. You see, in a live stream like this, where we are a little bit in time pressure, yeah, I sometimes have to do some shortcuts. It's kind of a little bit like a proof of concept, I would say. Okay, and now I'm going to do the following. You know what? I'm going to use another microscope slide here as well, because I do want to use different types of stains as well. So here and here. Some people asked me if I reuse the slides, and yes, I do reuse them. So again, not ideal, but then again, it doesn't have to be perfect for our purposes. As long as some parts, this one is not nice. As long as some parts are intact. And it's not even properly mounted to a table either, so it's a little bit improvised, I do admit. So, and I'm going to also now use this one over here. This is too thick. Okay. Maybe I should have checked it first before doing the others. So, this one actually, I think it was sliding over it. Yeah, this one did not really work well. It was too thin, so it was kind of sliding over it. And then let's give it a try. Yes, now I caught it again. So, okay, I'm going to now do the following. I'm just going to take this slide here, and we're just going to have a direct look under the microscope. No, but before I do that, let's have a look how the pollen is doing. Let's turn out. Yeah, they have notably grown pink. Okay. Yeah, so that's the pollen, yeah. Low frequency, yeah, about the yeah. But again, I somehow would have to get the microtome knife into vibration somehow. And I don't know how I would, yeah, I don't know, some kind of a piezo thing maybe. So let's have a look. Ooh, looks very thick. Not good because no water. And here we have to maybe, yeah, this is, go down with the, Ah, yes. So that's at low power. Here we go. Actually, it's quite okay. And when you see those plant, that's a monocot plant. So you see that the vascular bundles, the xylem and the phloem, the ones that are transporting the waters, are basically scattered throughout the stem. It's very different from a dicot like a sunflower. Some weeks ago, I've put a sunflower under the microscope as well, where those vascular bundles are arranged nicely in a circle. But in a monocot plant, the vascular bundles are all scattered throughout randomly, more or less. Yeah. Okay, I'm just reading commercial vibratomes operate at 100 hertz. Okay, that's fairly low frequency. That's a frequency that we could even hear. So let's have another look here. It's a slightly higher magnification. So and I would like to now try the following. I mean, that is now just as it is, without water, without anything. I mean, I could try it with, you know what? Let's try this one here with with water, just for the fun of it. Okay. We don't want to dry it out, so let's take a little bit of water. And I'm checking the time as well. I think it's fine. I always do a rough time planning, so then I'm able to show you the things I want to show you. And we're just going to add here a little bit of water. Of course, we add a small cover glass to see how this actually works. Okay. Yeah, it's fairly thick, but it's surrounded by water. So the refractive index difference is not quite as high, and it should look a little bit brighter. We're going to have a look. So where is the scope view? Yeah, exactly. It does look a little bit brighter. That's now basically with water as a mounting media. And you can actually see, look, if you actually, now I can also go up higher with the magnification. Okay, let's close the condenser a little bit. Okay, this is now a regular bright field, and by using the fine focus I can actually check how many cell layers I'm able to see. And you see there's only, we just look at one, let's just look at this one over here, this big cell, and let's focus all the way through it. It still stays bright, 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 bright. I don't see another cell going into focus. You see? And this basically means that the cross section was actually thinner than a cell. Because if this would go out of focus and another layer would go into focus, it would mean that the cross section is fairly thick. It's multiple cell layers. But if it stays basically like that, more or less, becomes blurry, then it actually means that the cross section is pretty thin. Yeah. So this is actually something that I sometimes do to make a little bit of a quality check. So going much thinner would not make a huge difference. So, but I would like to stain, okay? And do a little bit of staining. This is how it looks like again, okay? Look, yeah. Let's close this a little bit, yeah. So, and let's do a little bit of staining. And this means that I'm going to now try different stains, and this is going to be a challenge a little bit because, yeah, I'm going to do the following. I'm going to just use this here. I'm going to go with the most powerful stain, which is again, you don't see this, which is again the crystal violet. And over here. Then over here, I'm going to use a red stain, and it's called saffronin. That's another one. And what I need next is, and I don't have that, just a second, I need a beaker. Here we go. And I need some water. Just regular tap water. And in the meantime, I'm waiting another minute. Let's have a look at this one over here. Yeah. Yeah, it's already dried up. Why does this look so... Is this some kind of a reflection? Why does this look so bright yellow down here? It seems to be some kind of a... That's interesting. It seems to be some kind of a reflection, maybe. It looks almost like it's fluorescent, but actually it's not. Yeah. So, okay, in any case, you can see that, yeah, look at all those blue streaks here, right? Yeah, as the pigment is drawn up. Yeah. So, let's go back to the desk. So I think that should be enough. Now I'm going to do the following. Let's clean up a little bit here because I don't want to have too much. And I'm going to not only do that, I'm going to also use Löffler's solution. I hope I remember this. And Fuchsin. So, but let's do one after the other, otherwise I'm getting confused. So look at this. It's now stained. I need to take it and I need to wash it. And then let's hope that it's not too dark. Yeah, this looks very dark. Okay. So I'm now going, this looks very dark, but we're gonna see. So I'm gonna drop a cover glass on top. It's way too thick as well. I'm gonna do the same thing over here. Yeah, maybe not a good idea to mix it with the other one. Who cares? And here we go. And let's have a look if staining actually makes sense here or not. I think the first one is probably going to be way too dark. So, but we're going to see. Scope view. Here it is. Ah, yeah. 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 Ah, yeah, but I am still able to show you that what I wanted to show you. That's good. So it's actually, yes, very good. Even if it's fairly dark, there are a few things that I want to show you. And that's actually good. Let's have a look at this here specifically. You're probably going to notice that, of course, the cell walls are stained quite well blue. Then you're going to see that sometimes the cytoplasm, the inside of the cell, is also pinkish. Yep. And sometimes it's white. And you're probably also going to notice that specifically the larger cells over here, very often the larger ones are white, and more if the smaller ones are more pinkish. And the reason is that it's white because the cell was actually cut open and lost the cell contents. Therefore, it could not be stained. And those that stain pinkish actually are still mostly intact. So if I were to make a thinner cross section, then more of the cells would actually look white because they would not stain on the inside. Yeah. Basically, rinsing, yes, you're right. I did rinse it. It's just a comment here. I actually did rinse it just for a few seconds in this little jar over here, the beaker. Yeah, that's why it's yeah, maybe not enough. So now let's have a look at the one that I used with saffron in, the red one. Ooh, that's overexposed. So, okay. What if I rinse in alcohol? I did not try that. We can experiment. Yeah. So that is now the one in saffron in. Yeah, and you see that. Maybe you want to always look at a place that's not so thick. So maybe that's actually better here. Yeah. And here. So that is basically... Yeah, and I've... Can I'm just... look, I'm going to sidetrack now a little bit. Maybe some of you know that many years ago, and still am interested in amateur astronomy. So basically, it's a small telescope doing some sky gazing and so on. And yeah, still occasionally like to do that. Now with digital cameras, it's much easier nowadays to take pictures of stars. And I just saw a YouTube video yesterday where people who are into amateur astronomy, they made beautiful pictures of galaxies and so on. And then I realized something. Maybe amateur astronomy is so popular because you can make nice pictures. Seriously. And maybe that's maybe microscopy is a little bit, at least also maybe the way that I've been doing it very often. So I'm not even excluding myself here. I'm focused always on microorganisms, bacteria, microbes, whatever, fungi, mold, which I consider fascinating. But this is not something that all people like, you know. But nice pictures, that's something that many people can identify with. So maybe I should, yeah, not forget about making nice pictures as well. And sharing some nice images and cool things, you know, patterns of nature maybe, right? Yeah. So, yeah. Where's the astronomy channel then? That's a good one. I don't have one. I don't have one. I've been thinking, yeah, of course, about also picking up astronomy a little bit more, but time is limited, so I decided, okay, I'm going to stick with microscopes for now. Yeah. Yeah, and astronomy is easier. Yeah, I mean nowadays a friend of mine has got this computerized telescope. It's fascinating. You can completely control it on a tablet, and then you can say, "Oh, I want to see that galaxy. I want to see that star, whatever," and then it will go out there automatically, and it will take a picture of it, and you can actually see the picture live on your tablet. Yeah, it's so cool. And microscopy is also going to that direction with they call it virtual microscopy, which is also used then also in medicine and in training and so on. Yeah, so it's pretty fascinating. Yeah, yeah. But I have to tell you, a weekly astronomy cast. Yeah, but I'm not deep enough into astronomy now. Yeah. I did a couple of years ago. I mean, if you're interested, I'm not going to do an astronomy stream, obviously, but I did several years ago. Must have been 10, 15 years ago. I was able to take a picture of the Venus, the planet Venus passing in front of the sun. It was pretty nice. Yeah. So, but let's move on. We are not in astronomy right now. This is still a microscopy and hands-on biology channel. Yeah. So, and I would like to now try, the other ones here. So what I have over here is I would like to stain this one here now with Löffler's solution, which is methylene blue. I don't know. This is not a very nice specimen that I have here, but I'm going to give it a try in any case. And the last one is, how do you pronounce this in English? Fuchsin in German. Fuchsin, I guess. This is also the same stain that's it's the purple one also for pollen. Yeah, you see, it's kind of reddish purple. Yeah, not purple, actually more pinkish. It's not. This one is not fully covered. So let's do the following. The stain always has to fully cover the specimen. It's not too. Now here we go. Okay. Going to put this away so that I don't spill it. And I need another microscope slide, and then I'm checking. This seems to be still doing fine. I hope, at least. What time is it? Yeah, because in a couple of minutes after this one, I would like to completely change topics, and I would like to show you that what I found. And I hope that it's still here. It's still here. Good. Because I was quite happy. So let's do this now. I'm going to take this. Generally, methylene blue staining does not really need a washing. Okay, so it also looks a little bit brighter. It's not a very nice specimen here. It's torn apart. And this one over here also looks fairly bright, but this one I am going to rinse a little bit. This is pretty damaged already. Doesn't matter. All we need to have is a few, how do you say, intact cells. So here I'm going to add some water. So here we are going to add some water, and a cover glass also goes on top here. I still have not figured out something with the camera. I know obviously there's this rule you always have to keep eye contact with the audience. I get it. But the problem is the camera's in front on top of my monitor and I'm looking at my desk, so I don't know how to solve that problem. So let's have a look now again at the thing here. Now that is now with methylene blue, and let's see how this actually worked out. Ah. It actually also looks, yeah, it might look different because it doesn't stain quite as deeply, but maybe it also has to do something with the thickness of the cross section. Yeah, this looks pretty as well. Okay, yeah. So, so that is. So with fairly straightforward equipment, you can actually get quite nice results here. And again, as you've seen, there was no fancy special preparation. It was just live on this live stream. Yeah, and if you take a little bit more time with that, and if you're a little bit more patient maybe and have more trials, I'm quite sure that the results will be even better. Yeah. Yeah, so let's have a look at the other one, the red one over here. Okay. Ooh, this is very strange. Okay. This is a little bit too intense. Huh. Here maybe. Okay. Yeah, this is not so nice looking, but it has to do maybe also a little bit with the thickness of the specimen. But you know what? Let's go down with the magnification a little bit. Yeah. Here maybe. Here. Ah, here. Here it's better. Yeah. I mean, you get the idea. So it's also a little bit interesting that the red colors they seem stronger than others. I think this has to do also a little bit with the sensitivity of the digital camera to the different colors. Yeah. Yeah. So that's fuchsine, which is the same stain that is used to stain the pollen. Yeah. I'm reading now again. Yeah. 40 years ago, last use of fuchsine was hunting mycobacteria. Ah, according to Nielsen. So there, it's also used to mycobacteria. Okay, some of you have been doing a little bit of research here as well. Thank you. Yeah. Okay. Yeah, some astronomy comments here. The thing is then I stayed up very long nights. I used to do astrophotography even at the time. I used to do many years ago. I used to do black white film development and picture development photography. So I actually had my own improvised dark room as well. Improvised dark room. It was actually the bathroom that I used. Oh, here it looks better, right? So I took pictures of stars and so on. Yeah, and things are so much easier these days. Obviously, also with microscopy. I remember back 30 years ago, still at university, I wanted to buy myself a microscope, couldn't afford it. All very expensive stuff, and fancy equipment. And nowadays, you go on Amazon, and a few days later, you've got a microscope. It's so much easier. Yeah. So, yeah. So that's the fuchsine one. And now I've been basically talking approximately 1 hour. And what I'm going to do now is the following: I'm just going to make a cut, so to say, and I'm going to really radically change topics. Okay. So because I just want to show this to you as well, which was actually not planned. I mean, I did plan the flower, but the thing that I'm going to show you now, I did not plan. And let's put this away. You see, you got to be super careful with those stains because if they start to put them somewhere where they don't mess up my desk or my clothing or whatever. So let's put this away. So I was very happy with this here, and I hope it's still alive. I just want to show you the jar where I got it from. A couple of months ago, I've went to a local pond. Lake actually, and winter, I might even have made a live stream of this and some winter sample, right? That's it. I just collected whatever I could find and put it in here. And then today in the morning, I had a closer look and I could not believe it what I saw. I was really happy. And I immediately put it on a slide, no cover glass. And I'm just going to show you now what I found. Okay, so just going to show you now what I found. So it's a concave slide, yeah, no cover glass, no, that's important. I don't want to limit its movement. So, just a second. Where is it? Where is it? Where is it? Where is it? Can I find it? Here, that's the water. Nope. Here it is. Ooh, it kind of shrunk. A Hydra. This is a Hydra. And I don't know why it rolled up so much. It stretched out very long, up to a centimeter long. And you can actually see its tentacles. And I was really, really happy that I found this thing. And it's really flexible and it's able to change its shape. And I don't know why it rolled up this much now. Let me check here. Yeah. But it's still alive. No, it didn't roll up. It's standing upwards. That's the whole thing. It connected itself. It connected itself to the slide at the foot over here, right? Yeah, and it's kind of now standing upwards. Yeah. So, yeah, it's been a while that you ever since seen a Hydra, and the reason why it's been a while is because I didn't find one. So, but I was really happy, and for those of you who don't know what hydras are, they are basically related to the jellyfish, right? They are not filter feeders like sponges, but they're predators. They're pretty aggressive predators, scary, because with their tentacles, they're able to hunt for tiny water animals. The mouth is over here. The stomach is in here, right? And as you can see there, it's a super flexible organism. Yeah, a planarian. I yet have to find one. Okay, and you know what? Why not add a little color here? Okay, and what I would like to do now is the following. I'm going to try something very mean and nasty. Oh, look at this. Here, that's now with a 4 times objective. Yeah. It's very nice. I'm going to now try something very mean and nasty, which is, do I have this worm here still? Where's the worm? I want to feed it something, but I don't know where the worm is. Just a second. I've got an I've got more of these worms, so if I can't find it here, it doesn't matter. It's got more of those. Yeah, I don't know where it is. It crawled away maybe. Did it crawl away? No, it might be here. I don't know. In any case, let's behold the Hydra again a little bit. Now it's kind of standing upwards, towards the microscope. That's why I did not put on the cover glass. Let's see. Look, okay, I'm gonna put in the other slide in here, and I'm gonna try to find where the worm is. Here it is. How am I supposed to pick this guy up? Okay, let's try to pick it up somehow. I need more water. I might have picked it up. I don't know. We're gonna see. Ha! Here it is. I wonder if it's able to catch it. I don't know. Okay? It's also a little bit a question of luck whether it's actually gonna touch the Hydra. Let's take the other slide out. And we're gonna see. This is a little bit of a patience. No, please don't move into the wrong direction. Yeah, I wonder. Otherwise, I might catch a few more of these worms. What do they eat? They like to eat tiny water fleas. They eat worms. Apparently, they also eat Iolosoma worms, like this one over here. They basically are predators, so they're not feeding on just basically filter feeders like bacteria and so on, right? Yeah. And this is now a little bit a pity if it's not able to reach the Hydra. So maybe I have to remove a little bit of the water here to limit its movement. Yeah, see, it's moving away. But it's, I guess, a question of time until it reaches the Hydra again. Yeah. Let's take this away. So, please touch the tentacles. Yeah, a couple of years ago, I've actually added a little tiny copepod, which is a tiny water crustacean. It actually, yeah, caught it. I've been feeding it also some nematode worms. It also killed those and ate those. But I've never done, the Iolosoma worm before. Wherever it is now, here it is. Here, that's the water drop. Maybe the water drop is a little bit too big. You know what I'm going to do? I'm just going to take it away and I'm going to make the water drop a little bit smaller. But the problem that I have is I have a difficulty actually seeing the worm. I need a darker. Yeah, I have a problem seeing the worm because it's so tiny. Yeah. It's, you know what, we're just gonna be a little bit patient. Scope view. Here it is. It likes to be. Hydra is where? Here. Maybe I'm just going to try to catch a few more worms. I'm just gonna add a few more. Let's see. I've got the jar over here. And let's hope for the best. Yeah. Let's see where this one is. Yeah, I've got some difficulty finding it. If it doesn't work, then maybe I'm just going to reduce a little bit the water. Okay. Because this is a concave slide, if I reduce the water, then the water is going to flow to the center of the slide, and hopefully they're going to be closer together. Here it is. Okay. So. The worm is on the other side of the slide. So let's. So here is the Hydra. Yeah, the questions are arriving. Thank you very much. But I might have already answered most of them anyway. And I really hope that it's able to eat it. I don't know. You can see that due to the lack of water, it has a little bit of a difficulty moving. Here it is. Yeah. I might be, otherwise I'm just gonna try to push it towards it somehow. Where is this here? Oh, okay. Yeah, I don't know. We're just gonna wait a little bit, and if we're lucky, we're able to see something here. Yeah. Um, so, see, that's a little bit one of those typical demonstration cases. When you essentially try to demonstrate something, it does take a little bit of luck. Yeah. But over here, I'm just going to get the desk view. Oh yeah. Oh, it's moving away. Yeah. So the problem with the nematodes is as follows. I added some vinegar eels a couple of years ago to a Hydra, and the vinegar eels they're acidic, right? So I got a little bit of acid as well, vinegar, and the Hydra immediately died. So it's very pH sensitive, and for this reason I tried to use actually those Iolosoma worms, but what I could try is in any case because why not? I can still try to do it with some vinegar eels, and I'm going to try to clean away the how do you call it, the acid, okay, the vinegar. In order to do that, I have to and then I hope for the best that this doesn't kill the Hydra, because I do want to put the Hydra back, of course, again into the jar. No, please, 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 please touch the Hydra. Oh, this was some dust. Yeah. So let's try differently. I'm just gonna keep on this one here. I'm just going to pull out a couple of nematodes. And very quick check under the microscope now whether the nematodes are plenty. I think the nematodes are plenty. Okay, so you get the idea, right? So, but I cannot add that to the Hydra because the Hydra is gonna die because of the acid. Yeah. So, and I'm now going to wash this with a little bit of how do you call this, water. So I'm just going to leave the Hydra there now, and I'm just going to tell you what I'm doing. To this drop of vinegar, I'm adding a little bit of water. Okay, and I'm removing, yeah. So I'm just keep on rinsing the nematodes because I don't want too much acid, and then I'm just gonna hope for the best. And then, so I'm gonna add a lot of, careful, I'm gonna add a lot of worms now, okay? I'm gonna add a lot of worms now. Where is this? Here we go. And let's see. I would be very disappointed if it wouldn't catch them now, because some time ago when I've done this actually The Hydra was very hungry and immediately add a little bit more water maybe to give the Hydra a little bit more freedom of movement. And maybe it caught already some of them. I hope at least. Or not. That's the typical demonstration effect. Ah, but look. Is there something? No, unfortunately not. Because sometimes what happens is that the nematode worms get stuck to the tentacles. But maybe actually this, look what happened over here on the right side. Do you see this over here on the right side? Some of them might have already gotten stuck to the tentacles, to the stinging cells, and then the worms become paralyzed. Okay. It's quite interesting here. It's pretty overwhelmed. And then excuse me. And then what they usually do is they actually then will start to pull over the tentacles and actually eat up the worm. Yeah. It's massively overwhelmed now with yeah. I think it broke free again. Yeah. Yeah, I don't know which toxins it is, but they have got so basically I think they're called nematocysts, which are stinging cells, which kind of shoot out microscopic poisonous darts, and basically like the jellyfish. Yeah, that's why. And then they paralyze the prey, and then basically they stop moving and then they eat it. Yeah, but over here this is way too many worms now. Yeah. So the whole thing here, okay, here there's still, let's see if, are the darts visible? Yes, you gotta check a little bit. In the other channel where I could actually see once, I could really zoom in. I could not see the darts, but I could see the strings that were attached to it. This was actually quite well visible. Let's do it like this here. I'm going to turn up, yeah. Oh yeah. There is actually a video also where I I added a tiny, I think it was a copepod or something, and it actually also tried to kill it and eat it. Yeah, yeah. But this guy is being bounced around quite aggressively. You know what I'm gonna do later on is I'm just gonna put it back into the jar, but at least, yeah, I guess. It feels more attacked by the worms than it is able to attack the worms itself. Okay. Wow, this guy is totally overwhelmed. Yeah, I've actually seen there is another Hydra in this because what they do is they attach to the inside of the jar. So what you have to do is you have to go in with a pipette like the one that I have over here, which where I cut off the tip, right? And then you can carefully try to suck it out. Basically you put it on a microscope slide and you remove any excess water. Yeah. So this one, no, this was way too much. Okay, the Hydra is stressed out by so many worms. Yeah. The thing that I'm on a different note, the thing that I'm a little bit fascinated is that you see those vinegar eels basically are now in water, in pure water, right? So this means they're pretty tolerant to pretty big changes in pH. Yeah. So and I've also seen one Hydra once which started to reproduce. So it uses asexual reproduction a lot. So there's basically like a tiny Hydra growing out of the side of the main one. Yeah, it's called budding, which was also quite nice to observe. Yeah, too much of too many worms. Okay, yeah. So what I'm going to do now is okay. I'm just gonna quickly fly through some of the questions that you might have. But I guess I answered most of them already. You mentioned Elmer's glue would shrink when making a leaf impression. Is there any way I can prevent this? I just want to show you, at least with some leaves, this was an issue. Just a second. Yeah, this Elmer's glue, when you apply it to the bottom of a leaf, in some plants, especially those that are very waxy and hydrophobic on the bottom side, I had a problem actually making the glue stick. It kind of really dripped off. Yeah, it really dripped off, and that's something that. Let me refocus this again. Yeah, okay. And so when you apply some glue on the bottom side, I mean, I can show you over here. As we watch the Hydra being bounced around. If you apply a little bit of this Elmer's glue, on the bottom of a leaf, I have to open this first, of course. Remove the dry glue. Okay. Yeah, sometimes, I don't know if you're able to see it, if you're able to spread it apart, and then what I've seen sometimes is that it actually wouldn't stick there, but it would actually pull together and then simply drip off. I guess it depends a lot on the type of leaf that you're using, on how well it actually sticks to the glue. But I would say actually trial and error. Yeah, it would be the thing. Yeah. With some plants it actually worked, but with others not. Yeah. So I don't know. This Hydra, yeah, maybe should have not added too many worms here. Yeah. So a couple of other questions here. The question is what kind of glue works? Then it can't be just wood glue. Well, actually, yes, it is. I just used regular wood glue, and it has the advantage that it's fairly liquid, right? And it spreads well, and it separates quite well as well. Yeah. Animal brain cells. You want to see some animal brain cells? I'll have to pull out someone from permanent slides. Wouldn't ultrasound pop the cells? So that's the question was the following, because my suggestion before is to use some kind of an ultrasound vibrating microtome knife, and I'm cutting the cells open anyway. So yes, it's gonna break the cells. If there are these ultrasound devices that you can use to actually disinfect even sometimes, or break open the cells, so ultrasound can be used. However, in this case, I do want to break and cut the cells in any case. Yeah. Let's see. I think the movement maybe is also a little bit too fast, maybe for. Yeah. This could be. And rinsing might be needed. I did that. Okay. Okay. So, just saying, and we're going to, as we look at this, I'm going to have a look at if I can find some slide with brain cells, but don't be too don't have too high expectations. Okay, let's have a look. It's gonna be in one of those 3 boxes here. Nope, not here. Not here. Yeah. Okay. That's basically the cerebellum of a cat. Okay. Which is all the way in the back. Okay, so the cerebellum is responsible for motor coordination. It's a commercial slide, and basically you should be able to see some brain cells here. And let's have a look at a few brain cells, and then I think I'm just gonna call it a day for today. And yeah, here it is. Yeah, what I'm gonna do is I'm just going to carefully later on after the stream, I'm gonna pick off the Hydra and I'm gonna put it back into the jar. But we're gonna have a look at the thing here, the desk view. Yeah, so that's how the slide looks like. It's a commercial slide from a cat. And this is basically a part of the brain. And if we zoom in here, it's stained and everything, we should be able to see the nerve cells. But again, yeah, because yeah, you might be disappointed. Okay, just saying. Because they're so densely packed that it might be difficult to make out the individual cells. Yeah. So we can zoom in. Let's have a look at a brighter area. So that's a little bit the issue with the brain. Too many cells so close together that it's difficult to see the individual cells. But you can actually see that there are these long, thin fibrous structures which might appear to be the axons. Yeah, because, yeah. So let's go up yet further. Yep. So all of those fine lines that you see here, right? Yeah, these are the nerve fibers. But the individual nerve cells are so densely packed together. Maybe a nerve cell body over here, yeah, with the axons and the dendrites. And these are basically the extensions stretching out. Yeah, but as you see, that's basically, yeah, brain of a cat. And just by looking at it, we can see how many cells there are. Yeah, different layers. And here, different parts of the brain look different because the different types of cells present. Yeah. We're again at lower magnification and again 40 times. Yeah. Yeah. So okay, just let's have a closer look again at the how the Hydra is doing, and then I think I'm just going to call it a day for today again. Wherever it is. Oops, it's way too dark, too bright. I mean. Oh, look at this. Ah. Yeah. Hydra a little bit overwhelmed still. Yeah. Yeah, yeah, I got a couple of slices of a cat tongue. I've got a rabbit tongue somewhere in the box. But maybe I'm going to keep that for next time. Which with the barbs on the surface. Ah, yeah. Yeah. The gastro trick. Yes. Occasionally, you have to be also lucky to find those. Occasionally, I also find those, but not quite as common as some of the other organisms that we have here. Yeah, I think I'm just going to call it a day today, and I'm just going to leave this here now, and I'm gonna take off the Hydra and put it back into the jar, maybe together with a couple of other nematode worms. Maybe they're going to be also a food for the Hydra in there. But I think I'm going to call it a quits for today again. Yeah. And I'm just reading the last comments. We can get around arranging another dual stream. That's generally also something that we have tried once. If to join in, maybe if you have also other things that you want to present or so, please feel free to send me pictures or so, or maybe to do some kind of a double live stream would be quite nice to try this again. In any case, I'm going to call it a day. At least I was able to show you a Hydra, even if it did not catch anything today. But maybe I'm able to still videotape something, and then I'll put it again also into this channel. I wish you all the best. Have a nice weekend. Happy micro hunting as always, and hope to see you again. See you around. Bye bye.